Podcasting from Lund, Sweden, the birthplace of Ingmar Bergman's man crush, Max von Sydow, the home of an unexplainable amount of handball teams, and one of the most advanced laboratories in the world, Max 4. This is Intronauts, bringing you inside the Max 4 facility, where you get to meet some of the greatest minds and the explorers of tomorrow. I am your host, Thånell.
And I'm your co-host, Laura Roster. Today we're excited to chat with Karina Thånell and Uwe Müller about some of the exciting research that goes on here at Max 4. So Karina, you are the project leader for Softimax Beamline and group manager for the imaging beamlines. What does that mean?
The imaging beamline at the moment consists of 3 beamlines, so 3 different stations where you can measure very, very different things. One of them is Nanomax. It's a hard X-ray beamline, and it's a microscopy beamline. So we look at very, very small things with different techniques. Then the second beamline is Max Beam, which is a different kettle of fish, you could say. Because there we use the photons, and these are soft X-ray photons, so they will not penetrate your teeth and body. And there we use these photons to create electrons that we then map in different ways onto a detector, giving us more electronic information. And so you have to think about materials that physicists get excited about, like superconductors and graphene and stuff like that. And then the last beamline that I have in the portfolio is Softimax. And as we said, I'm also the project leader for that. And the reason for that is that one is not yet up and running, but we will very soon be there. So within, a couple of months, we hope to take first light and then take it from there. It's also, as the name implies, a soft X-ray beamline, so more of a gentle stroking of the materials, you could say. And here we are going to look at different kind of materials. It can be anything from environmental science, soil particles, or roots of plants, for instance, or seeds, to biological things, tissues or cells, to even also kind of physicsy things. So graphene or small pieces of batteries and stuff. That's what I do.
Wow, so nice to have you here, Karina. And it sounds like you have a really broad portfolio here at Max 4 working
with. Yes, you could say. I mean, of course, there's a lot of people working in the team that do the actual work, and they are much more experienced in the techniques of the particular beamlines, of course. So I'm mostly there to keep a little bit of the overview, let's say, and hopefully point all our noses in the right direction. But they do the actual science a little bit more than I do.
Right, so cool. So can you start by telling me a bit about your background? Where do you come from?
Well, I was born in Holland, or the Netherlands, as it is properly called. And I studied there as well. I studied physics because, from an early age, I was always intrigued by getting to know more about how things work and why things are the way they are. And you can't get more fundamental than, the building blocks of the universe, basically. So, physics it had to be.
If you think about this curiosity that you've had, is there a childhood story to tell in there where you saw that in yourself?
I don't know. I think as a child, you don't really realize that because you think everybody is like that. So I don't know if there's one particular story. It's just that I've always looked at things and then really have the kind of head tilt and then think how does that work? So I mean, from early on, I could think about if you see clouds in the sky, that you think what's holding that up? In the sense why don't they just drop down, or sort of is it really fluffy inside, or could you stand on it or something like that? I was very small at that time, obviously. And then the fascination also with astronomy. I mean, looking at stars and seeing all these things with the spiral galaxies and these beautiful pictures that were coming out of NASA and the Voyager series. So I mean, there's always been this kind of interest in reading about it or finding out more about it, and it's not really a particular thing. It's always there. I mean, as soon as I look at something, then it's usually there's a question of but why and how. And after my PhD, which I also did there, I moved to England for a while to do a postdoc, as it's called. And there I got introduced to working at synchrotrons. And one of the synchrotrons that we visited was MaxLab in the old days, so the predecessor of Max 4, which was closer to the center in Lund. And once when I was there doing experiments, I talked to one of the beamline scientists and said, like, it would be really nice to work in a facility like this. And MaxLab was one of my favorites because there you could just ring the doorbell and be let in. It was sort of like a friendly atmosphere. And he tipped me about a position that was open at one of the beamlines, to which I applied, and the same evening got a reply, when can you start? So, and the rest is history, as I say.
Touching onto that, I think a lot of the research that goes on here at Max 4, for an outsider, it can seem quite abstract. It can seem that there's no clear connection between, doing this kind of imaging or using this kind of equipment and finding out why clouds don't drop from the sky or why the universe works the way it does. Yeah. So how do you find that connection or what sort of keeps you excited and keeps you coming back here?
Yeah. Well, obviously, I went a little bit beyond the clouds kind of thing. You soon figure out that it's just water vapor. But so if you nowadays, if you come across a problem, because in the meantime, of course, you've gathered some knowledge. I studied physics and I've studied a lot of different topics, worked with different people and techniques. So you have in your head, you have a little bit of a portfolio, as you will, of different ways of looking at things. If you talk to the different people at the different beamlines who are experts in their technique, and you know a little bit about the physics or the chemistry of a particular thing, then it's quite easy to set up an experiment where you say, like, well, I've always wondered about this little thing. So, if you have a battery and you want to know why the efficiency goes down after a while, you might sort of like with somebody who is in spectroscopy, so who can look at chemistry of things, sit down, set up an experiment and say, look, okay, if we do it like this, then we can rule out that it's not like this, it's not like that. But you sort of make the experiment suit your question, as it were.
So these questions, they just form in your mind when you're looking at something else, or?
Yeah, exactly. It's sort of like, you read an article, you discuss things with somebody else, or it's based on what you have done before, or you see something unusual in a particular experiment that you have done that you at first cannot explain. And one of my favorite things is a good interview with the Nobel Prize winner André Geim, who got the Nobel Prize for graphene, this two-dimensional sheet of carbon chicken wire. And he said that in his group, what they used to do is on Fridays, they sit around, drink maybe a couple of beers, and discuss new ideas. And these new ideas were at the base of finally observing graphene. So they were sitting and discussing this, making it from a 3D to a 2D kind of material and seeing what the properties were. But then, of course, you need a proper 2D material. Nature doesn't always want to play ball, so you have to convince it to get there. And they said, well, if you take graphite, it's already a layered material. So why would it not be possible to peel off just one layer and then study it? And then, of course, the question is like, well, how would you do this? And the solution was surprisingly simple. Scotch tape is all they needed, basically. So, with just sitting and discussing with your colleagues some crazy ideas, good things can come. Mm. Obviously, he also said, like, we discussed a number of other ideas that we tried and didn't go anywhere. It's not always bingo. But a Nobel Prize is not bad for a couple of Friday afternoons, I would say.
So you can come into science looking at it from this kind of curiosity way of just trying to understand how the world works and just looking at the world and trying to understand it. But there's also this constant question that you get, I guess, that is, what can it be used for?
What is it good for? Yes.
Yeah, how can this be applied?
Yes. Yeah, and to be brutally honest, personally, I am not so interested in the end what you can do with it. And that's because the way I am, you could say. Other people, like my husband, for instance, if we sit on the couch watching television and we come across Discovery Channel, he'll get excited if there's big megastructures being moved around or sort of like a ship being built of gigantic proportions or technical complexity. whereas that usually makes me do, food preparation or something. And on the other hand, if there's something that is more deeply about explaining things in nature, like in galaxies or through the wormhole with Morgan Freeman, then that makes him do the cooking, whilst I sit there transfixed to the fundamental explanations of things in nature. So it depends a little bit on how you're built, I would say. And most of these things that we do, it might not have a very clear purpose from the outset. But if you think about the fact that we have computers everywhere and that we have chips in our mobiles and can do many, many more things than we could like millions of years ago, all of this research has come because somebody at some point looked at a fundamental property of a material and thought like, okay, how does that work? And then once they understood a little bit more about how does it work, somebody else usually came along and said like, oh, but this can be used for something. So, I mean, by us giving people one piece of the puzzle by saying it's like, okay, so this is as much as we know how it works. Then somebody else can say like, okay, but then I know if I make it into this kind of structure and connect it to something else, then I can get it to do something really wonderful that is good for you. And, I wish them luck with that.
And I know you're just starting out with a project that I find super fascinating, and that is about those sea squirts. So those are a special type of animals that they start out almost like little fishes, like they have a vertebrae almost, and they're kind of like us. And then they settle down onto the seafloor and they become almost like plants, like they're sedentary, they lose their nervous system, and then they just spend their life on the ocean floor. And why are you so interested in these curious animals?
To say I'm working with that is really pushing the thing, because I mean all that has happened was basically one of these, let's say, Friday afternoon, although it was over coffee, chats. Because I was actually working on something else. I had been looking at data taken at Nanomics, where you can see metals in a nano pattern on the surface, different metals. And I looked at which metals are important in biology on Google, just to see, because I mean everybody knows there's iron in blood, there's magnesium in photosynthesis cells in plants, and you have calcium in your bones. But what other metals are actually used by biology? And then lo and behold, I found that vanadium, although we don't really need it as humans, but vanadium is used by these sea squirts. And that got me a little bit intrigued, because vanadium is like, it's an odd element to use. And these sea squirts, they concentrate the vanadium from the seawater. So in the seawater, it's maybe like, you have milligrams or something per liter, whereas in their bodies, they concentrate it 1 million times. So that got me thinking, like, what do they do with it then? It was discovered in 1911 or something like that, so around about a century ago. And then since then, people have looked at it a little bit, but it's still not entirely sure what they use it for. So, it only grows in curiosity. It's like, what are these animals doing with it? Because vanadium is a very nice metal. It can exist in different oxidation states. You can make a battery out of it. So, I mean, you can completely out of vanadium-containing compounds. No, normally you have like two, right? I mean, you have a copper-zinc battery or something like that. So the one side is copper, the other side is zinc with an electrolyte in between, and electrons flow hither and thither. But with the vanadium one, you can do it with only vanadium, because it can do vanadium 2+, 3+, 4+, 5+. So you can have multiple states. So one of the things that I'm thinking at the moment is indeed that there is some sort of energy effect, so that they store it in a particular form, and then when they need the energy for whatever reason, they release and sort of like switch an oxidation state to free up the electron and give the energy. Which is not solely my idea. I mean, it's not just that there's also some research that points at that thing.
So looking at those fundamental questions about these animals, how can Max 4 help you explore that?
Here at Max 4, we have a couple of beamlines where this could be studied quite nicely. So at Nanomics, for instance, one of the techniques is to map out the elements that are present on a very, very tiny scale. So we can have a resolution of about, 50 to 100 nanometers. And a nanometer, just to give you an idea, is 1 billionth of a meter, which maybe still sounds very abstract. But if you have a marble and you said, like, if this marble was the size of a nanometer, then on that scale, the Earth would be a meter. So, really, really small. So we can look at maps of distribution of different metals and also sulfur and phosphor, for instance, in different specimens. And in this case, you could take a slice of C² and look at where the vanadium is. And even if you play your cards right with the energy, you can have a look at what kind of oxidation state it is in. So that is very cool. In SoftyMax, we will be able to do the same thing and very, very much more easily look at the chemistry of things. So really to figure out what kind of surroundings the vanadium ion has. And this can also be done on Balder, by the way, another beamline that is not in my portfolio. But they also have a very, very sensitive way of doing this in a slightly bigger specimen, basically.
So tell me a little bit of what kind of results you get out of that or would get out of that. Do you get like actual images? Or?
Yes, exactly. You would get actual images, and then depending on which beamline you would use, you would see, for instance, in Nanomax, you get the vanadium map, maybe not the oxidation state so easily, but you would get to see where the vanadium is alongside other metals. So you could also very easily see the iron, if they have any manganese, sulfur, phosphor, anything like that. Whereas at SoftyMax, you would get similar maps, but then you can look at the oxidation species. So you get a very, very detailed map. These blood cells where the vanadium is located, they are on the order of 5 to 10 microns or so. So they're very small. And for the bulk spectroscopy, you could take an entire C², as it were, maybe grind it down a bit. Sorry, animal lovers. And then look with Balder, who is very, very good. It's another beamline, who is very, very good at doing the nitty-gritty spectroscopy, where you really get information about the direct surroundings of the vanadium even. And I mean, of course, this is my personal little interest. We have a whole bunch of users that come with different questions, and they also come to use our beamlines for their research. I mean, it may be batteries, it may be catalyst, it may be fundamental physics, or it may be proteins. I mean, anything, as long as they can formulate, maybe together with us, the right questions. Then we're here to help answer. And for me, it's a good source of ideas, and also it adds to my toolbox, because sometimes users come with new ways of making samples or looking at things that you thought, hey, that is not something I could do before. So it's very stimulating to live in an environment where people from all sides come in and do their research. So, I mean, in principle, that is the continuous drive, let's say, to do research and to know more.
Thank you so much, Karina. It's been really, really nice chatting with you and hearing about how to explore these really fundamental questions.
Yeah, thanks.
So now we're joined by Uwe Müller. So nice to have you here.
Thank you.
You are the group manager for macromolecular X-ray crystallography. So what does that job entail?
Yeah, I mean, you talked about more or less two jobs. One is to be the group manager, which means I take care as a line manager for the group of experts like scientists, engineers, which try to support and develop the macromolecular crystallography beamline BioMax. In addition, I'm also a scientist and try to use these beamlines to support our users and try to do some own research activities at Max4.
So you have some projects that are really looking into using Max4 to create things and to make things. Can you tell me a bit more about that?
Yes. We at BioMax are concentrating on very complex matter, namely biomolecules, proteins, for instance. These are the molecules where all organisms are made of, and they determine everything, the life, the reproduction, developments of all organisms. And we want to know the three-dimensional shape of all these proteins, because the shape is related to its function. And we want to know what are they doing, how they do that, and maybe even how one could influence this function. For instance, if you want to develop new drugs for fighting diseases.
And how do you look into those details? Like what exactly about the protein that we want to see?
So we can see objects or atoms in these molecules which are extremely close to each other, like 0.1 nanometers, which is 0.1 times to 10 to the minus 9 meter. And it's very hard to imagine how small that is, but we see single atoms with this microscope.
How does looking at proteins and molecules at this really fundamental level, how does that translate into new medicines?
We are using an approach which is called rational drug discovery, where we first try to understand how the new medicine, we call it drug molecules, are interacting with proteins, which we call target proteins. Those are molecules which we know are involved in diseases, and these small molecules are binding to these target proteins and then, for instance, prevent some functions. They block these proteins and then something cannot happen, which is then beneficial for healing this disease. And this rational part in this is that we improve our molecules and then do it again and again until we reach certain properties of these interactions. And in addition, we created recently a project at Biomax, which we call Fragmax. And with this project, we are able to do the search for a potential drug molecule very efficiently. And here we have we call it library of small molecules, these so-called fragments. And we throw all these molecules on our protein molecule. And you have to imagine this is like millions of little drones which fly over this protein in a very short period of time, like 5 minutes. And if they find something where they, for instance, can bind, then we can observe this event. And from there, we can then design new fragments, let them grow a little bit, let them become bigger or more specific, and then we do the test again. And with the hope that the interaction, for instance, is then enhanced.
And the binding is sort of when the medicine works or when something happens in the body.
Exactly. So only if we have a stable binding of our medicine, then this has an influence on the organism. Yeah.
So it's almost like you're playing Tetris with molecules and trying to move them around in different ways and reshape them so that they fit together better.
Yes. And with X4, which is currently the most intense X-ray light source from this technology point of view in the world, we can do it very efficiently. So for instance, we can test, say, 200 of these fragments during one working day against a protein and get a lot of structure information. Yeah.
So when you're investigating with this microscope and the different beamlines, would you say that you produce an image? Can we see this, or is it something different?
Yeah, at the end of our investigation, we have a so-called three-dimensional map of the electron density distribution in our sample. It's not a single protein molecule, but a so-called protein crystal. So we build up a three-dimensional object out of our molecules, which is a crystal. And within this object, we can then see all the atoms, how they are arranged to each other, and how our little fragment molecules are binding to this protein. And this is then an image which we can, for instance, visualize on a computer. We can turn it, we can fly through this molecule, we can manipulate that whatever we want, and look at this as scientists and try to understand how the interaction is made of. And
So all of these different tests of seeing how biological molecules interact with each other, are there any specific diseases where you have seen results or that you're looking into right now?
This is actually a task mainly for our users, especially the pharmaceutical companies. So all Swedish pharmaceutical companies are coming to Max4 to do these investigations at our beamline, and they don't tell us what they are doing. So the answer is yes, we do work on this task every week, but I cannot tell you what these diseases are they are working on, because they don't share this very secret information with us.
So thinking about all these amazing questions and these things that we can tackle at Max4, what would you say the hope for the future research in life science will be, or the big ideas?
Yeah, I mean, life science plays a huge and growing role at such a facility like Max4. And the dream here is to have experiments and environments where we can see directly into living organisms. We want to see how a cell, for instance, works in a living organism, and understand on a very the holistic view on these complex parts of this organism, how that works. And then we can combine these information with other methods which we are using to get a big picture how the whole organism works, so to say. This is a dream, and it's not easy, obviously.
But I'm really looking forward to seeing that dream come true. Thank you so much, Uwe. It's been really fascinating to talk to you.
Yeah, it was a pleasure.
Thank you.
So we've been listening to two very different researchers today.
Yes, definitely. I mean, you have the fundamental on one side and the applied science on the other.
Yeah, and it's really interesting that there's so much going on both these types of research, both these sides of the coin, the more fundamental research and the more applied science here at Max4.
Yes, and there are examples now, but even more I'm looking forward to hearing.
Thanks a million for listening to Intronauts with me, Thånell Kornfeldt, and Laura Rasta. If you like the show, please leave us a nice review on iTunes.
And don't forget to tune in for our next episode when we visit a beamline and talk to scientist Nuna Prisla about her research on climate change.